So today I'm going to answer some questions about exome sequencing. Exome sequencing is a question we get all the time as a genetic testing company. So, in the realm of genetics and genomics, exome sequencing is one of the most powerful tools we have diagnostically to look at the genetic code of an individual. The exome allows us to uncover disease causing mutations and unravel the mysteries of genetic disorders for diagnostic purposes. So, what is exome sequencing? Well, the human genome, which is the essentially the language that makes us who we are, consists of 3.2 billion base pairs. This is representing about 19 to 20,000 genes. You've probably heard that much of this is junk DNA. That's not quite true, but only a small fraction of these genes, roughly 1 to 2%, are responsible for making the proteins that make us who we are, which are the functional units of the human body. So the represents the portion of the genome that consists of these protein coding genes. So it's about 2% of the entire genome. So exome sequencing is a technique where we selectively sequence just the exome or that 2%, which is important, or the one that encodes for protein. The reason we do this is it's a lot more complex to sequence the entire genome and much more expensive. In contrast, the exome sequence we can do in a relatively short period of time. And importantly, it has most of the disease causing variants that have been identified today. So, how does exome sequencing work? Well, first we collect a sample. The typical sample is an individual's DNA. This can be collected from blood, saliva, a buccal swab, or many other sample types. What we do in the laboratory is we isolate that DNA using specialized laboratory techniques, and then we make a library. You may have heard of NGS panels. This is essentially where instead of looking at the entire exome library, we look at very specific genes related to a disease process, like connective tissue disease or intellectual disability, for example. We then take that library and we sequence it on a number of instruments that are designed for next generation sequencing. And then the hard part comes. So even though we have the sequence of the exome, we then have to do a bioinformatics process to look at the generated DNA sequence. We compare it or we align it to a reference genome, and then we use our bioinformatic tools to identify genetic variations. These genetic variations may just represent the difference between two individuals, or they may represent disease-causing um, variants. We then look at these variants or these genetic alterations, and we look across databases to see if they actually have been reported to have clinical significance, if they will change the structure of the protein, function of the protein, etc. So, what is whole exome sequencing used for? Most importantly, we use it for genetic disease diagnosis. Exome sequencing has proven particularly valuable in diagnosing rare genetic disorders that are especially challenging to identify using traditional diagnostic approaches. By identifying disease causing mutations, exome sequencing helps doctors provide accurate diagnoses for rare genetic diseases and also allows us to facilitate targeted treatment, for example, in the case of cancer. Uh, which is another broad area. So, in addition to rare disease diagnostics, we use this frequently in cancer genomics. Exome sequencing plays a vital role in understanding the genetic basis of various cancers. By analyzing the tumor sample and comparing them to normal tissue, we can identify the mutations that then identify the physiologic mechanisms involved in that cancer development and progression. Now, a lot of therapy is personalized to these specific pathways, and this allows inclusion in clinical trials for targeted therapies. We also can use the exome for pharmacogenomics. In our laboratory, we use a different process for pharmacogenomic analysis. And then, importantly, we can use exome sequencing for research and discovery. So, exome sequencing has emerged as a transformative technology in the field of genetics and diagnostics. Its ability to selectively sequence the protein coding regions of the genome allows us to have insights into the genetic basis of disease. Um, we have a frequently asked questions about exome sequencing on our website. We offer whole exome sequencing as well as NGS panels for a variety of tests, which can be found on our website.